everyone. Today we'll be discussing Introduction to Molecular Diagnostics. So this will be the last topic for cytogenetics. It referred to as the detection of various pathogenic mutations in the DNA or RNA, and in order to facilitate detection, diagnosis, subclassification, prognosis, and monitoring response to therapy. So it is used to expand scientific knowledge of the natural history of diseases, identify people who are at risk, and diagnose human diseases at a nucleic acid level. So it combines with the laboratory medicine with the knowledge and technology of the molecular genetics. So the information revolution in molecular biology is permeating every aspect of medical practice. So molecular understanding of disease is translated into diagnostic testing, therapeutics, and eventually preventive therapies. So to introduce essential concepts in the molecular diagnostics that impact on the identification of the novel markers of human diseases. So it is used to develop and apply useful molecular assays to monitor disease, determine appropriate treatment strategies, and predict disease outcomes. So what is the history of the molecular diagnostics? So in 1865, Gregor Mendel, The Law of Heredity. 1866, Johann Michener, The Purification of DNA. 1949, sickle cell anemia mutation was first studied. Then in 1953, the structure of DNA was discovered. 1970, recombinant DNA technology was discovered. DNA sequencing in 1977. PCR, or the polymerase chain reaction, was discovered in 1985. And the Human Genome Project started last 2001. So first, the sickle cell anemia, so a genetic disease caused by a single nucleotide change on the 6th amino acid of the peptide chain of the hemoglobin. So Linus Pauling introduced the term molecular disease in the medical vocabulary based on the discovery that a single amino acid change leads to sickle cell anemia. Then James Watson and Francis Crick on the structure of DNA was published. It is one of the most important biological discovery in the 20th century. Then discovery of the DNA structure, 1955, 12 scientists received Nobel Prize. Then 1958, Frederick Sanger, the insulin sequence. Then 1962, the DNA structure. Then 1984, monoclonal antibodies. So the first seeds of molecular diagnostics were provided in the early days of recombinant DNA technology. So complementary DNA cloning and sequencing were invaluable tools for providing basic knowledge on the primary sequence of various genes. So DNA sequencing provided a number of DNA probes allowing the analysis by Southern blotting of genomic regions, leading to the concept and application of RFLP, or the restriction fragment length polymorphism. So Kary Mullis, 1985, he invented the PCR, and then in 1993 he received the Nobel Prize. The unusual origins of the PCR, 1993. So PCR revolution. So PCR has greatly facilitated and revolutionized molecular diagnostics. So the most powerful feature is the large amount of copies of the target sequence generated by exponent amplification, which allows identification of a known mutation within a single. So PCR markedly decreased need for radioactivity. So it either used for generation of DNA fragments to be analyzed or is part of the detection method. So the Human Genome Project that started on 2001. So its goal is to identify all genes in the human DNA, determine sequences of 3 billion bases that make up human DNA, create databases, develop tools for data analysis, and address ethical, legal, and social issues that arise from the genome research. So impact on human diseases. So discover potential novel molecular markers of human diseases. So identification of novel molecular markers of human diseases and utility of molecular markers to develop useful molecular assays for detection, diagnosis, and prediction of disease outcomes. So we need to monitor diseases more accurately. It can be used to determine most appropriate treatment and reduce patients' morbidity and mortality. So practical application, diagnostic. For the identification of disease, prognostic outcome of a disease, predictive possibility of a disease, and therapeutic, the response of a disease to treatment. So on molecular pathology, we have the hematology, identity testing, genetic diseases, solid tumors, and infectious diseases. So on molecular genetics, we have single gene disorder, polygenic disorders, and chromosomal disorder. On molecular oncology, diagnostic testing, disease prognosis, and determination of predisposition. In hemapathology, diagnostic testing and determination of clonality, identity testing is parentage and clinical testing. Infectious diseases, qualitative and quantitative detection of infectious agents, microbial identity, and genotyping and the drug resistant testing. So basis for technology, advance in the understanding of structures and chemistry of nucleic acids have facilitated the development of technologies that can be employed effectively in molecular diagnostics. So the molecular technologies in the clinical laboratory, first we have the amplification technique. So that involves PCR, the polymerase chain reaction. We also have LCR or the ligase chain reaction, and the NASBA, the nucleic acid sequence based amplification. We also have the DNA sequencing. Hybridization technique, we have the southern, the northern, and the western blot. Electrophoretic methods, we have the SCCP or the single strand conformation polymorphism or the DGGE or the denaturing gradient gel electrophoresis. We also have recombinant DNA technology and the biochip technology that uses DNA microarray and protein microarray. So for the nucleic acids that are targeted by the molecular assay, so genetically based diseases can be diagnosed, specificity can be controlled, single base changes can be detected, and expression of gene is not required, targets can be amplified. So cause or the etiology, mechanism or the pathogenesis, structural alterations or the morphologic or the molecular, and then the functional consequences or the clinical significance. So pathogenesis, understanding the molecular pathogenesis of human disease enables an effective utilization of molecular assays. So in the diagnostic, distinguishing variants of human disease based on the presence of specific molecular markers. Prognostics naman, prediction of likely patients' outcomes. Therapeutic, I response to specific therapies based on the presence of a specific molecular markers. Then the genetic lesions in the human disease, identification of genetic markers, identification of disease-related genes and molecular targets for assay development. Characterization of gene sequences facilitates characterization of disease-causing mutations and molecular targets for assay development. Beneficial outcomes from the Human Genome Project, improvements in medicine, microbial genome research, DNA forensics, improved agriculture and livestock, better understanding of evolution, and more accurate risk assessment. Human Gene Project, the ethical, legal, and social implications, use of genetic information, privacy, psychological impact, genetic testing, reproductive issues, education standard and quality control, commercialization, and conceptual philosophical implications. So what's great about it? So as many as 5,000 diseases have direct genetic causes. So high sensitivity and increased specificity for tests adds diagnostic utility. So potential for simple standardized procedure and automation. So rapid throughput or throughout, increased number of techniques for infectious diseases, a viable reflex for equivocal morphology, and price are falling. So the ultimate goal of molecular diagnostics is to provide molecular information that will combine with and complement information related to patient history and symptoms. So it is also used to provide more sensitive, precise, and accurate determination of disease diagnosis toward appropriate and effective treatment options.